Okay. Well, maybe no one heard that, but on Zoom, but that's fine. But Andy is this year's winner, and then next year it's going to be Shauna Newton. Shauna's doing a work with Kelly Branch on SCAD and CT scan, so we're going to be excited to hear what she's doing next year. But I'm going to hand this to Francis to talk to Andy, or talk about Andy.
Okay, thank you. So it's my pleasure to introduce Andy for this year's Laughlin Award winner. Andy is a native of New York. He did his undergraduate work at University of Rochester, where he majored in bioengineering. He went to Maryland for his MD and PhD, and he worked for Dr. Lederer, who's one of the leaders in calcium imaging and signaling. We were very fortunate at the University of Washington to successfully recruit him to come here as a resident. I know he was heavily recruited amongst other programs, but he did his medicine residency and short-tracked into cardiology and has been involved in research for the past 2 years. He's the author of over a dozen papers. He's the only person I know that's batted 1,000 in terms of getting grant funding. He was successful for getting a T32, F32, AHA, and then the Washington Research Foundation Award. So he's been very successful in obtaining money, which is a primo example of someone who'll be very successful in academics. So I'm very happy to introduce Andy, and we look forward to your talk.
Okay. All good on Zoom in terms of hearing and seeing. Okay. Great. Well, thank you very much, Francis, and happy to be here and share some of the work that I've been doing in the Murray lab. My project's titled Arrhythmogenic Calcium Signaling in Cardiac Stem Cell Therapy. So this will be kind of a fun exercise for our group. It's a little more of the kind of basic translational lab bench research, but mixed in with a true translational objective in terms of regenerating the heart. And so this is work that I do in the Murray lab with my mentor, Chuck Murray, and also work that, as Francis said, is supported by the Washington Research Foundation too. And so I have, let me see. Okay, I don't have any disclosures today to share. So the outline for today's talk, we're just going to introduce some key aspects to stem cell biology and how this forms the basis for our therapy for heart regeneration. Discuss briefly some of the ongoing clinical trials that are underway for bringing cardiac stem cell therapy to patients. And these are very early phase 1 trials, so it'll kind of just be introducing the groups. And then one of the key aspects that we worry about with this therapy is arrhythmia. And so we're going to go over some of the data that we've generated here at UW related to cardiac arrhythmias after cell therapy. And then my work looking at calcium as a potential molecular driver of these arrhythmias before we finish up. And so heart disease, as we know, is a leading cause of death worldwide. And the AHA just recently put out new statistics in terms of the burden of cardiovascular disease in America, and it's quite high. If you include all forms of cardiovascular disease, roughly 50% of American adults will have some form. And coronary heart disease affects almost 5% of adults in our country, with 800,000 people having a heart attack each year. So it's a massively important aspect of health in our country. And with that carries a large economic burden. So the AHA released the kind of estimates and predictions for growth in terms of the economic burden of cardiovascular disease. And it's expected to almost triple by 2050. So really astronomically high numbers that go towards our healthcare, all related to the heart. And so what we see as kind of a next step in kind of elevating the care that we can deliver to patients is creating a platform by which we can regenerate the heart after ischemic injury. And so the idea being that you can have a thrombotic event in a coronary artery, And that will lead to an injury in the downstream myocardium. And over time, this injured myocardium will remodel and the hearts will become weaker and potentially the patients may undergo heart failure. And the idea is that this injured myocardium will scar. And in order to prevent this, we have one pillar of our therapy is to go in there and open up the vessel as soon as possible. And that's called revascularization. We can use medications to do this, stents, or bypass grafts. And then we have very good medications to go and try to prevent the remodeling, the negative remodeling of the heart after injury. So medications that target the neurohormonal axes in our body, as well as some interventions like mechanical interventions and resynchronization therapies that can help with this process too. But currently, we don't have a pathway to regenerate the heart. There's no drugs, no treatments that will allow the myocardium that dies, which after a heart attack, we think is almost 1 billion cardiomyocytes are dying. We don't have a pathway by which we can replenish those cells yet. And we know that the human heart has a pretty limited regenerative capacity intrinsically. And so there was a very, very clever study done in 2009 where they looked at the amount of carbon-14 in the atmosphere. And you can see this surge in carbon-14 in the atmosphere in the mid-1950s, and that correlates with actually nuclear bomb testing. And so what they were able to do is basically, and then after 1963 with the limited test ban treaty, you see this precipitous decline in the carbon-14 in the atmosphere. And so what these authors were able to do is correlate the amount of carbon-14 that accumulated in the DNA of heart muscle cells from people born either kind of before this surge or during this, and get an idea or an estimate of how frequently these cells are being turned over. So a really clever use of kind of atmospheric and just kind of broad science, bringing that to the cellular level. And what they found is only 1% or less of cardiomyocytes turn over. And so over the course of our lifetime, only 50% of these cells may turn over. So really kind of quite you're almost born with what you end up dying with. So it's a really interesting path. And in our group, in the Murray lab, we look to use human pluripotent stem cell derived cardiomyocytes to rebuild or remuscularize the heart. And this is really the passion and the life's work of my mentor, Dr. Murray here. And the idea being that we can take pluripotent stem cells, cells that can be turned into any lineage of cells, and give them chemicals and molecules to direct their differentiation into cardiac muscle cells. And our hope and our goal is to use this to create a dose of cells that we can then deliver to patients' myocardium to replenish that muscle. And the idea being you take this injured myocardium and you rebuild it, you remuscularize that. Over time, we've become very efficient in generating these heart muscle cells. So originally, we used growth factors that were kind of the intrinsic signals in the body to direct this differentiation of the stem cell into the cardiomyocyte. Now we can use small molecules that target a specific signaling pathway called the Wnt signaling pathway. And we can activate this pathway and then inactivate this pathway at a very timed sequence to generate these cells. And so we can do this in culture to make these beautiful sheets of beating muscle cells. And with a single plate, we can get about 15 million of these cells. We can also do this in suspension, where we can use bioreactors to generate over 150 million of these cardiomyocytes. And so we have a couple of different pathways which allow us to create different scales of cells for the goal of studying them and for heart regeneration purposes. And so about 15 years ago, our group started using these cells to remuscularize animal models of heart attack. And so we started with a rodent model, a rat model, where we can take these cells and inject them into an animal after giving that animal a heart attack, a myocardial infarction. And what you're seeing here are graphs of these cells inside, surrounded by scar tissue, so injured myocardium. And what we were able to find is that when we do echocardiograms on these animals after 4 weeks following the transplant, they have an improved function in terms of their their fractional shortening doesn't decline any further from the time point that they get the cells. And they have structural remodeling with limitation in the dilation of the heart, as well as thickening of the heart muscle wall. So things that you would really want to see in a clinical therapy for this. And if we fast forward about 10 years from that study, over this time period, our group became extremely adept at producing large quantities of these cells at the scale that you would be able to remuscularize a larger animal. In this case, what you're looking at is tissue sections from non-human primate. And in the top, you can see in blue the scar tissue and the thinning of the heart muscle wall after a heart attack. But in the bottom panel, you can see these green pockets of cells, which are actually the human pluripotent cardiomyocytes that we transplanted in this. And most importantly, if you look at the trace here, we can measure the ejection fraction, so the signal of how strong is the heart pump function at different time points. So before the heart attack, right after the heart attack at the time point where we're giving the cells, and then 1 and 2 month, 3 month after giving cells. And in blue are the animals that received the cardiomyocytes, and then in sham, the squares, they did not receive that. So a pretty remarkable that, in both groups, you have a relatively similar drop in the heart pumping function, but the animals that received the cardiomyocyte injections returned to near baseline. So this is kind of some of the most exciting data that you can really find, and led to what's now underway, which is a number of clinical trials testing this therapy in human beings. And so these trials are in very early phase 1 of recruitment. So almost none of them have recruited more than 20 or so patients. The first one in this list here is out of Stanford by Dr. Wu's group, and it's sponsored by the California Institute of Regenerative Medicine. And they're using cardiac catheters to deliver different doses of cells to patients that have ischemic cardiomyopathy, so patients who have heart failure after having a heart attack. There's the Help Therapeutics group, which is based out of China, that has a number of studies ongoing that are looking to deliver these cells in the context of going into our coronary artery bypass surgery, so CABG surgery, as well as transcatheter delivery of different doses of cells. Similarly, there's a group out of Japan, the Heartseed program, that, as far as we know in terms of what was published, did the first in-human injection of these cells back in March of last year, I believe, or March or February of last year. And they're using, again, allogenic stem cell-derived cardiomyocytes that are cultured in this spheroid pattern and this kind of specialized needle adapter to try to deliver the cells in a robust manner to the myocardium that's injured. There's a German group that creates almost like a cardiac patch of cells and implants them on top of the injured myocardium, which is that's kind of the one study ongoing using that pathway. And then there's a spin-out HeartWorks from the Mayo Clinic that is using autologous stem cell-derived cardiomyocytes, so taking cells from a patient and inducing them into becoming pluripotent stem cells, and then trying to regenerate and reconstruct our myocardium that has congenital anomalies. So really interesting work on multiple fronts. But one of the things that we're most eager to hear about, because these data related to these studies hasn't really been published yet. We know that anecdotally that patients that receive these cells actually do undergo arrhythmia. And that's not necessarily surprising. Back in 2014, when our group did the first large animal study with pluripotent stem cell cardiomyocyte transplant, we actually saw that the animals went into a fairly robust, the primates went into a fairly robust arrhythmia. So they transitioned from sinus rhythm to AIVR, which is kind of almost like a rate-controlled ventricular tachycardia. but some would go into ventricular tachycardia, often not in the primates transitioning to ventricular fibrillation. So mostly tolerating this rapid rhythm, but still a very scary thing if we saw this clinically. Our group further went on to kind of map out where this arrhythmia comes from. So what you're looking at here is an electrical mapping study done by Steven Sessler, one of the electrophysiologists at Seattle Children's, collaborating with our group. And so you can see that the red signal originates right from this point, and then transitions out into the rest of the myocardium. And so that suggests that this is a focal origin of the arrhythmia. So a focal origin being where we put those cells in. When we use different types of EP testing to do overdrive pacing or cardioversion to try to stop the arrhythmia, it basically goes right back into it almost instantaneously. So it didn't work. And so that suggests again that this is a focal triggered arrhythmia from the cells we put in and not kind of the classic reentrant arrhythmia that we see most commonly in patients with ischemic heart disease. And then we couldn't induce this arrhythmia either with a programmed electrical stimulation. Now, we also know that this is a transient process. So the animals, the large animals that get this arrhythmia, they don't have it forever. They only have it for a period of time after the transplant. And so whether we give the stem cell cardiomyocytes derived from humans into monkeys, or make monkey stem cell derived cardiomyocytes and give them back to a monkey, or use the human cells in a pig model, each of these animals, each of these large scale models gets the arrhythmia. But if we look at these graphs here, the arrhythmia burden in terms of hours per day or percent of time in the ventricular tachycardia is limited to just about 30 to 60 days. So we know that we really have this window that we need to kind of protect people within and get them through before these graphs likely mature into a form where they're stable and less likely to cause problems. Another really wonderful study that was done by our group with Dr. Nakamura from our cardiology division and Dr. Murray was looking at, can we use antiarrhythmic drugs to help with this process? And we found that we can. So while the antiarrhythmics, amiodarone, which is a class 3 antiarrhythmic, but is really broadly active, and then in combination with ivabradine, which specifically targets the channel responsible for automaticity, the channel responsible for what's called funny current. And so if we use this combination of drugs, we can drop the burden of arrhythmia down by 30%. But perhaps more importantly, we also drop the heart rate of these animals from 150 or more beats per minute in the non-treatment group down to a more tolerable 120 beats per minute. And with that, the animals survive. So none of these animals died of the cardiac arrhythmia or cardiac death. So really kind of an impressive thing that's clinically important and from our understanding, clinically in use now with these current trials. And so we know that the immature physiology of these stem cell derived cardiomyocytes is something that's promoting the automaticity. And so what you're seeing here is a diagram of an action potential from these stem cell derived cardiomyocytes. And you can see the morphology compared to that of a diagram of an adult cardiomyocyte is far different. These these cells have the presence of phase 4 depolarization. So these funny currents that kind of slowly trigger the automaticity of the cells. And they operate at a membrane potential that is more depolarized or closer to thresholding than their adult counterparts. And so we hypothesized that the automaticity of these immature grafts leads to the arrhythmia, and that the maturation of the cells after they're transplanted will play a key role in limiting this process. And we can see this maturation if we, on the transcriptional level, so the gene expression level, using our rat model. So in this study, we were able to take our rat model of a heart attack with cell transplant and sacrifice the animals at different time points to harvest their cardiac tissue and do sectioning. And from those sections, we can actually capture, using a laser dissection, regions of the graft and take that little region of graft and measure the gene expression profile in them. So really, really clever work. And looking at those differential gene expressions, we can see that in the first week after transplant, almost 1,000 genes change whether going up or down. So really a ton of activity at the cellular level. And that's shown here in this volcano plot, where we're looking at kind of statistical probability on the y-axis and then fold change on the x-axis. So basically the dots highlighted in blue are statistically downregulated, or the dots in red, highlighted in red, are genes that were overexpressed in the transplant tissue. And so if we look at days 7 to 1 month after transplant, you have a huge number, again, of genes that are changing, almost 1,000 genes, many of which are calcium handling genes or critical electrophysiology genes. But then after that point, very few genes change, only like 25. So this first month is this kind of window of at least genetic or gene expression maturation. And this was information that was used to kind of guide one of the most recent papers from our group that was led by Dr. Marciano and Dr. Murray. And this was using a CRISPR-Cas9 gene editing strategy to try to prevent this arrhythmia. And so what we did was we took stem cell-derived cardiomyocytes and we created a population of them that had a few genes or knocked out or overexpressed, so edited. This included knocking out, getting rid of that funny current responsible for that automaticity depolarizing portion of the action potential, as well as the T-type calcium channel, which also plays a role in that phase. And then overexpressing a potassium channel, KCNJ2, which is responsible for repolarization and maintaining a more negative membrane potential to create something that's a more mature a phenotype for these cells. And so when we did this, these cells they went from being spontaneously active and depolarizing to being quiescent, but we could trigger them to contract. When they were transplanted into a pig model, the wild type cells were underwent engraftment arrhythmia, while the edited cells did not. But there's one other key edit that was done in this study, and that's knocking out the sodium calcium exchanger. So the sodium calcium exchanger is a transporter on the cell membrane that's responsible for moving calcium from inside of the cell to outside of the cell. And it does so by transporting 1 calcium ion out and bringing in 3 sodium ions. And so there's a net charge difference there where there's 3 positive charges coming in and only 2 positive charges going out. And that leads to a depolarizing current. So the potential for after depolarizations or triggered activity. That's the kind of the foundational component of a calcium-dependent arrhythmia. And that led us to hypothesize when I first talked with Dr. Murray about joining his lab, that perhaps excessive calcium leak from the intracellular calcium store in the cell, the sarcoplasmic reticulum, is playing a big role in these stem cell-derived cardiomyocytes to promote their automaticity and after transplant, the engraftment arrhythmia. So just to briefly introduce how is calcium important in our heart muscle cells. This is a diagram that's probably one of the most famous diagrams related to cardiac excitation contraction coupling, where you can see the cell membrane with the sodium calcium exchanger, the L-type calcium channel, these kind of invaginations of the membrane deep into the cell, which are called transverse tubules. And what happens is the electrical action potential that's at the cell membrane triggers activity of these L-type calcium channels to stimulate a large release of calcium from the SR compartment. That's called calcium-induced calcium release. And this chemical calcium signal is what's sensed by our contractile elements in the heart muscle cells to cause a contraction in force generation. So at the cellular level, cells contract, which you can see here in this myocyte that's been loaded with a calcium indicator dye. So it shortens after it's stimulated, and there's a big flash of fluorescence, which is a flash of calcium. And that calcium is what drives activation of contractile elements for force production and is critical for pushing and moving the blood out of our heart into the rest of our body. Now, what is calcium leak? Well, if we look at another video of an adult muscle cell from a rat, again loaded with a calcium indicator dye, you can see these brief flashes of fluorescence if you look carefully. So right there was one, there was one. And these flashes of fluorescence are localized calcium release events from clusters of ryanidine receptors, the channels responsible for calcium release. And they're called calcium sparks. Now, the structure of adult cells has these clusters of ryanidine receptors spatially isolated to prevent multiple sparks from activating all at once. However, when we depolarize the cell during an action potential, we can coordinate this activity. Experimentally, we often will use what's called line scan imaging to rapidly measure just a small region of these cells over time. And so for a calcium transient, we would be scanning in space, the same small area of space over time, and creating these chymographs of the calcium signal. And you can see the global calcium transient here. Similarly, if we were just looking at spontaneous activity, sparks would look something like this. And this is kind of, these calcium sparks were actually discovered by my PhD advisor at the University of Maryland, Dr. John Lederer. So this kind of physiology at the cellular level is something that I've been part of for a long time, and it's kind of near and dear to my heart. So it was kind of a real pleasure to meet with Chuck and see that he was interested in applying these skill sets and this physiology to the stem cell and the engraftment arrhythmia level. But how are we going to do that? So we ended up using a genetically encoded calcium indicator. It's called GCAMP. You can see the structure here that has a green fluorescent protein that changes intensity of fluorescence when it's bound to calcium. And so the Conklin lab at UCSF created a stable expression, stably expressing stem cell line, the WTC11 stem cell line, that stably expressed GCAMP6F. And so that's what you're seeing here is these flashes of fluorescence and this green signal being the calcium rising and falling in the cell. So when we started working on this, we wanted to say, hey, let's work in a system where we can see that, make sure that we can see a difference using these cells. And so down here, you're seeing chymographs. They're pseudo-colored for just kind of increasing the clarity and contrast here. And so you'll see some pseudo-colored images throughout. But what you're looking at here is the calcium transients from cells that were cryopreserved and then thawed. And so in black, they were cryopreserved and thawed just 3 days before doing the imaging, while in blue, they were given about 2 weeks to recover. And so you can see there's a much more robust calcium transient once we give these cells 2 weeks to recover. And so some of the metrics, we're going to just have relatively similar analyses and slides kind of throughout the remainder of the talk. So one of the metrics that we look at is the amplitude of this calcium signal. So how big is it? What's the peak compared to the minima here? And so that's calcium transient amplitude measured in F over F naught, so normalizing the fluorescence intensity. And in this case, we did get a larger calcium transient. We can also look at the kinetics of the calcium. So how fast does the calcium rise in the cell? And so that's a measure of the time to peak for the signal. And then we can look at how fast the calcium declines in the cell, or the kinetics of decline. And we can see that in terms of time to 90% relaxation for the cells. And so the kinetics are markedly adjusted in these stem cell populations that have been allowed to recover for 2 weeks after being cryopreserved compared to those that were immediately imaged. We can also use these same use cells that were never preserved, but just kind of day 20 early cardiomyocytes, and look at how they respond to fight or flight signals, so beta adrenergic signaling. So classically, and so what you're seeing here in black trace is the average transient from about 15 cells that were before they were treated with isoproterenol, or the beta adrenergic signal. And then the red trace being the fight or flight signal. And you might wonder, that looks kind of odd. Well, in an adult muscle cell, we would expect the calcium transient to go from to become larger and faster. So inotropic and lusotropic effects in response to fight or flight signals. Now, these immature cardiomyocytes, they don't have the cellular machinery to act that way. So what we actually see is perhaps even a slight reduction in the calcium transient amplitude, likely due to increased leak of these ryanidine receptors after the isoproterenol. And the kinetics don't really change too much. Again, the cellular machinery is not advanced to the point that it can do the adult kind of physiologic changes. But what we did see is if we stop the pacing, you have increased spontaneous activity of the cells. So that's what you're seeing here and quantified there. So while there's not the inotropic and kind of the adult fight or flight response that we would expect for a fully mature cell, these immature cells do have what's called a chronotropic response, or they beat faster in response to the stimulation. So with this kind of ability to do the imaging and skill set in hand, we wanted to ask a more difficult question. How does the calcium handling in stem cell derived cardiomyocytes change after they're transplanted? So what happens, how do the cells mature in vivo? And there's really only been one study that's done this in a non-injury model, just in kind of a neonatal bioreactor model, where they took stem cells from either mice or from humans and turned them into cardiomyocytes and transplanted them into neonatal rats. And they were able to show that there's increase in the maturation of the calcium handling, the gene expression analysis, things of that nature. But this isn't really the same as what we anticipate for human beings that had a heart attack. And so we wanted to use a different animal model. We wanted to go back to our rodent model, our rat heart attack model, where we take a rat and it undergoes an ischemia reperfusion injury for an hour. We close it back up and 4 days, allow it to recover for 4 days before we transplant 10 million of these GCAMP encoded cardiomyocytes into the heart. And so that's what you're seeing here are day 20 cardiomyocytes, so just a few days after transplant. And then what we're able to do is re-isolate the heart out and then use a kind of a classic Langendorf retrograde perfusion to dissociate the cells from those hearts. And so here what you're seeing are cells that were actually inside of the animal for 2 weeks. And so we can then take these cells and do calcium imaging on them. And so that's what you're seeing in these traces here are the global calcium signals from in vivo matured stem cell derived cardiomyocytes. And so again, we have our chymographs with the day 20 in vitro cells on top and the in vivo cell on the bottom as a representative, and the average global calcium transient in green for the in vivo matured. And so you can very clearly see the rapid kinetics. And so we were able to show kind of an increase in the amplitude and a marked change in the kinetics, of far faster kinetics, more mature handling of the calcium in these in vivo matured cells. Similarly, they actually didn't have as much spontaneous activity as their in vitro counterparts. Now, the problem with this, though, is I can only find these cells when they're actually contracting and blinking, when there's activity going on. Otherwise, it's kind of a Where's Waldo situation. And it's, I don't have a mechanism of seeing the cells, and that limited our ability for throughput with this. And so what we ended up doing was creating our own cell line. So we went back and we actually took a cell line that was made in South Lake Union at the Allen Institute that expresses a red fluorescent protein on the cell membrane. And we then were able to edit these cells to insert kind of the newest variant of GCAMP, GCAMP8F. And so it's slightly faster, better kinetics than the prior one. And so we can, again, make these cells. And so now we have our WTC11 cell line that's red all the time, so we can easily find it, but has our calcium sensor so we can do these calcium-dependent experiments. So really kind of a critical feature. And the workflow for our study for in vivo maturation is mostly as follows. So again, we're using our ischemia reperfusion model, giving the animals 4 days to recover, injecting 10 million cells, and then allowing them to mature in the animal for a period of time before we re-isolate the hearts or re-isolate the cells. And we can plate them down for calcium imaging or fix the cells for downstream experiments. And so we plan to do the following groups. We're looking at the cells kind of right around the time of transplant, and then we want to see how they change functionally. So 1 week after engraftment, 4 weeks after engraftment, with key experiments being the calcium signaling, staining of the cell kind of ultrastructure, the potentially proteomics or some form of omics on these isolated cells we have, and then we can do some tissue staining of the frozen heart blocks as well. And so I'm going to show a little data from, we kind of worked our way from the longest time point back to try to give ourselves the most mature cells at this time. And so I'm going to show some data looking at our cells re-isolated from 4 weeks in the confocal imaging there. And so actually the cell that you've already seen is one that I was able to re-isolate out of a heart after being engrafted for 4 weeks. And I put it through a protocol where I would pace the cell at half a hertz, so 1 beat every 2 seconds, or 1 beat every second, and then 1 beat every second in the presence of these fight or flight chemicals. And what we would expect if these cells were fully mature is that as we transition from half a hertz to 1 hertz, there should be a positive or a larger calcium transient, so a positive calcium frequency relationship. Similarly, as before, when we add isopaternal or fight or flight signals on, we would expect a larger and a faster calcium transient. But what we ended up seeing was slightly different. So there wasn't a huge difference when we did this paired analysis of 12 cells that we were able to re-isolate out. So they're still pretty hard to find, but we can find them. The calcium transients don't really get markedly bigger, maybe slightly larger with the isopaternal. And so that's quantified in our calcium transient amplitude plots here, blue being half a hertz, black being 1 hertz, red being with isopaternal. Our time to peak for those groups and our time to recovery, really not huge differences. But one of the things that was really cool that I found when I re-isolated these cells out was sometimes you would see cells, but many times you would see these clusters or basically pieces of graphs that we're able to take out. And so that's what you're seeing here. And so these are all individual cells that are kind of coalesced and interconnected, being paced at 1 hertz and then 1 hertz in the presence of isopaternal. And with that, we can actually get a whole lot more cells. So from 20 clusters that I was able to isolate out and find, we can have about 80 cells or so that we can go through our analysis there. And so when we look at the isopaternal response with that in a paired fashion, we actually do see a larger calcium transient there, but it's still not as fast or as kind of robust as you would expect in a fully mature adult cell. We have to remember these cells have only been in an animal for 1 month. And an adult cell has been maturing its whole life there. So it's kind of a stepwise process. And this is also an injured host. And what I'm really excited about next is for these same cells and cell clusters, I not only measured the global calcium trigger signals, but I also measured the spontaneous activity of these cells. And so what you're seeing here are cells that are not stimulated, but these flashes of fluorescence are flashes of calcium release that could potentially trigger an arrhythmia. And I'm really eager to further quantify this, and I'm working on that now to kind of calculate out how many of these calcium release events are occurring, what their kinetics are. And for the clusters, I think we can also get some interesting data related to how these cells are interconnected, which could be really important for understanding the substrate for arrhythmia in these graphs. And so just to summarize the calcium data that we've gone through here, we know that genetically encoded calcium sensors now can really facilitate and bring ease to these calcium signaling studies compared to using dyes like we do for most adult heart muscle cell studies. Cryoinjury or cryopreservation, I should say, requires time to recover for these cells. And early on, the fight or flight signal seems to be fully limited to chronotropy or how fast the cells will pace. As we mature the cells in vivo, though, even after just 2 weeks, we can see some markedly mature calcium dynamics. And that's something that I'm eager to see when I go back and do my in vitro cohort for the new cell lines that I produced. But these cells still lack a fully mature phenotype. So, they do not have the anticipated kind of calcium frequency response or adrenergic or full adrenergic response after 4 weeks being in vivo. And it's going to be, in terms of connecting this to the arrhythmia, it's the spontaneous activity that's kind of the danger zone. And so we want to be able to quantify this and characterize this in great detail so that we can really understand the substrate that's causing arrhythmia for these patients and identify new mechanisms to target that. And so just kind of concluding on this, the cardiac stem cell-based therapies, they hold huge clinical potential. We have 20 years of data from University of Washington now that shows that we can improve cardiac function with these cells. And the first round of trials are really building off of technology that was developed here. So it's kind of a really exciting time to see what happens in these trials. And the early anecdotes suggest that even at just getting 50 million cells, so remember I told you that 1 billion cells die during a heart attack. Well, now even when we only deliver 50 million to patients, we've heard that they still undergo arrhythmia. And these cells are much smaller, probably almost 5 to 10 to 1 for a cardiomyocyte. So just a small amount of cells can really potentially cause a problem. So it'll be important to see how the severity of this arrhythmia occurs in human beings to inform how we direct the generation of kind of second generation cell therapies targeting cellular automaticity and calcium dependent processes. And so the mechanistic bench work that I'm doing in the Murray lab is critical for characterizing these pathways and identifying where we need to intervene. And so my future work over this next year of research that I have is finishing the in vitro and in vivo maturation time points, and then building upon this functional studies to kind of further our story there and understanding of this process of in vivo maturation. And then we have another project that I'm not talking about today that's related to a stress signaling kinase, CAM kinase, and engraftment arrhythmia. And my goal is to have this work be published and to kind of set the foundation for my career development and kind of moving forward as eventually an attending physician, but then also someone with a basic science, translational science lab. And so I'm really grateful to a lot of folks. Specifically, I'll start with Dr. Murray. I mean, I don't think I've ever had a meeting with someone who, on our first meeting, basically looked at what I'm capable of doing, looked at what they're doing in their group, and said, "Hey, let's team up and let's take your skill set and merge it with my skill set, teach you a lot of great stem cell biology, transplant biology, kind of translational cardiovascular research, and merge it with the biophysics that I had for my PhD." So I think it's been an extremely fun process learning how to do that. And I think that's really what my goal with a postdoc has been. Similarly, Chuck's put together a phenomenal team here. And so Sylvia and Elahe really too have kind of held my hand in this introduction to molecular biology for me. As you know, a biophysics guy, we didn't really do that in my old lab. And we have wonderful surgeon in the lab, Leslie Blakely, and great staff scientists, Hans and Shulan, and great students and research staff like Bonnie and our students, Zhao and Nora, that have been working on this project with me. So really appreciate all their kind of help and support. And then the division has just been extremely supportive to me. So Dr. McClellan has really kind of held the way in kind of facilitating my career development within cardiology. Kenta, who's here today too, really helped me a lot with putting this talk together, going through it with me and kind of getting that ready. And then from just career development standpoint and being a physician scientist standpoint, Fareed and Francis and Kelly have been kind of excellent mentors. So I really do appreciate that. And you know, we have a neighboring lab, the Yang lab, which you know, Daniel and his team are just wonderful colleagues to work with. So the heart regeneration program has been really fruitful in terms of teaching me the stem cell biology. And then the Institute for Stem Cell and Regenerative Medicine, the imaging core, Dale Haley, has been incredibly helpful in getting this live cell imaging up and running. So really grateful to the Laughlin family and the support that you guys have provided for kind of pushing research here at UW and UW Cardiology to give us the opportunity to do fun experiments that hopefully will be able to impact patients down the line, and then other funding from the Washington Research Foundation and RT32 and things. So we'll take some questions.
Thank you, Andy, for a wonderful talk. Let me ask you kind of a clinical question first. You talked early on about some phase 1 clinical trials with these stem cells. So is the data sufficient now to recommend that these people get amiodarone and anti-therapy during this maturation phase? Is that part of their protocol, or is that?
Yeah, so different studies have different protocols, but pretty much all are using the work that that was started here and using amiodarone and ivabradine at times to kind of make this as safe as a therapy as possible. But they still do have the arrhythmia, yeah. The hope is that they tolerate the arrhythmia in a way similar to the non-human primates tolerate it well, whereas other animals such as pigs actually don't tolerate the arrhythmia well. And the structure of those animals are different. And so humans are closer to primates. So our hope is that these early trials go well and facilitate kind of further growth in this field.
Questions from the audience? Jordan?
That was really great. Really interesting and fabulous work that you guys are doing. My question has to do with heterogeneity. So you talk about injecting these 50 million cells, and I'm sort of thinking geographically. Is there a difference between the center of that engraftment and more on the border zone in terms of any of these calcium properties that you've been describing?
Yeah, I think there would be. So we kind of know from a lot of the prior work that from our group that the areas of the grafts that integrate with the host mature better. They mature faster. And so one would expect that they have different dynamics than the grafts deep within the host. And I think what I'm seeing is that when I isolate out these cells, first off, they're being injected into the injured portion of the myocardium. So they have limited blood flow to those regions. Kind of when I'm dissociating them and using enzymes, that enzyme is probably not really getting into the heart of these grafts, which is why I see these big clusters there at times, which is somewhat advantageous. Now, the single cells perhaps are coming from kind of closer to that edge or nearer myocardium that may have slightly different dynamics. But in the actual animal itself, I would definitely anticipate that there'd be heterogeneity, and that heterogeneity could in fact actually support almost like a intragraft reentry event, you could imagine.
Andy, that was just a fantastic talk. Just following up on Jordan's question, when I was seeing those clusters, I guess I was thinking, do you think that maybe Langendorf is not the correct way to do this? What do you think of doing cardiac slices? Is that possible? Because maybe the spatial, relationship is kind of important, because with Langendorf, you're biasing. That's probably the reason that you are seeing that early cells is that you're biasing against getting some of those cells out there, maybe are rogue and not with the cluster. Can you comment about that, or what do you think about using cardiac slices?
So I think that what you're highlighting is why kind of in cardiac biophysics, we do a multi-scale approach usually. And so we actually do have capacity to do cardiac slices. The cardiac slices, you'll kind of get gross, I think you can get gross kind of graft calcium dynamics, but you're not going to get as much of the subcellular activity that we can get in these kind of single isolations, because they're thick. It'd be really difficult to take a thick section like that and do confocal imaging and imaging at kind of fast resolution to kind of to get those signals. So I think it depends on the question that you're asking. And certainly there's a role for slice and a role for things like optical mapping to do this all at different scales, and how that all integrates together is really important.
Thank you.
Thank you, Andy, for a great talk. I was wondering, have there been other mechanisms of arrhythmia that have been seen with these stem cells?
Yeah, so that's a really good point, and something that Kenta and I were actually talking about a bit yesterday. I think that it is very possible that the primary substrate for the AIVR or the ventricular tachycardia is different than perhaps the substrate for the ventricular fibrillation, because not every animal that goes into the VT will degenerate into VF and die. In fact, the primates generally don't. And so it's possible that the automaticity component is really critical for triggering that VT. And then this is still a scarred myocardium, as kind of Jordan brought up. This is still an area where there's heterogeneity within the graft that can promote kind of changes in how these cells are repolarizing over time and kind of facilitate different substrates for arrhythmia. That's something that, I think from our standpoint in terms of the electrical mapping, but then also the work that Sylvia did, kind of stopping automaticity and really largely stopping the arrhythmia, leads us to think that the primary process is automaticity, but certainly secondary processes, and maybe during times of kind of degeneration to VF, it's a coalescence of this automaticity and maybe some facets of reentry too.
All right, thank you very much.
Yeah.
Yeah, I think we had a question online. Rob asks, great talk. Congratulations on your great talk. But he's wondering about the effect of controlling automaticity on maturation.
Yeah, that's a really interesting point. And it's this idea that perhaps the cells need to go through this phase of maturation after transplant to get to their final endpoint of being able to help support improved function in the host myocardium. I think that's something that we just need to learn more about, and it's certainly a good hypothesis. On the flip side of that, though, we know clinically that patients who have significant burden of ectopy, ventricular ectopy, have a high risk for heart failure and negative remodeling that is directly related to that ectopy. And so, one would imagine that after this period of VT, or just kind of this engraftment arrhythmia, the cells should be even better and even more functional. But it's unclear how important that phase is to maturing the cells, and it very well could be.
There's another question from Thompson. Is there a maturation effect in the normal embryonic development? Is there arrhythmogenic tendency in a normal embryo?
So, the fetal hearts beat very quickly. So while our resting heart rates as adults is more like 60 beats per minute, I think many of us who have gone in to see ultrasounds of kind of our developing children, and you can hear the heartbeat from Doppler, you can see it on the ultrasound, and it goes really fast. I mean, it goes 150 beats at some phases, it's 170 beats per minute. But it's not necessarily an arrhythmia. It's just kind of I wouldn't call it an arrhythmia. I would just call it the intrinsic rate of those cells. And so when you're going that fast at your baseline, it's unlikely to have kind of further arrhythmia on top of that would be really challenging. So I don't know if that answers the question fully, but I think in the process of going from kind of fetal or young heart tissue into kind of more mature and adult tissue, there's this kind of dynamic change in the beating rate that's kind of intrinsic to the maturation process.
I was curious, during the maturation phase, does it matter what the underlying pathology is, whether it's ischemia versus fibrosis or inflammatory cardiomyopathy? Does the original injury insult to the myocardium affect the maturation phase during transplant?
That's a good question. I don't know that we fully studied that, but I know that when we can take these cells and transplant them into uninjured hosts, and they still get engraftment arrhythmia, and they still take time to mature. Is that maturation faster in the uninjured host? I think one would probably hypothesize that it would be, because the injury is only coming from kind of the injection points of giving the cells, and otherwise the myocardium should have a robust capillary network, and blood flow to those cells should perhaps be a little bit better. So it could be a dynamic difference there.
Well, thank you, Andy, again for the wonderful talk. It's a fitting end to our Grand Round season. So this will end for this year, and we'll see everybody again next September.